Okay, my name is Aimee Ryan. I'm an associate professor in the departments of human genetics and pediatrics at McGill University and a member of the Center for Research in Reproduction and Development. What I want to talk to you today about is some of the research that's going on in my lab. We are particularly interested in studying epithelial cells and epithelial cell layers during embryonic development and how they behave. Epithelial cells are held together by tight junctions that form at the top of those epithelial cells, and we're studying the proteins that are involved in that process. One of those proteins that's essential is this small, relatively small protein called claudins. There are 20 family members, and there are about 200 amino acids. They have 4 critical domains. One is the transmembrane domains that localize them to the plasma membrane. They have 2 extracellular loops that determine the size and the charge of molecules that can move between epithelial cells, and they have a cytoplasmic tail that acts as a docking site for adapter proteins and small molecules that cycle between the cytoplasm and the tight junction. And this can regulate the interactions at the tight junction. We are particularly interested in how this family of proteins behave in embryonic development, and the processes that we're most interested in are the asymmetric positioning and the formation of organs, neural tube closure, and kidney development. As well, we are studying how the tight junctions regulate the migration of germ cells into the testes and into the stem cell niche where they develop. Of these, heart development, which requires asymmetric organogenesis, as well as neural tube closure, represent the 2 most common sites of defects during embryonic development. So neural tube closure occurs in a stereotypic fashion between species, and these steps are outlined here. The first step is that the cells at the midline must undergo apical constriction to allow that epithelial layer to bend. There are then forces from the side that push the sides of the neural folds together, and ultimately you get a closed neural tube and an overlying layer of ectoderm. We have shown that claudins are essential for this process. So there's a number of claudins that are expressed during neural tube closure. They have slightly different expression patterns, and we've shown here, I'm illustrating 3 types of claudins, blue, green, and black. If we remove the green and the blue claudins, we find that we block neural tube closure, and we don't get these cell shape changes that are important for the morphogenesis. We are currently screening patient cohorts to identify mutations in claudins and looking at where those mutations are located, and then trying to understand how they affect the protein function. In particular, we are looking at, do these mutations affect neural tube closure? Do they affect how organs develop and how they determine if they're going to be positioned on the left and the right side? And do they affect nephrogenesis in the developing kidney? By understanding this process and understanding these variants and what they do to disrupt the function of claudins, we can understand some of the proteins that are involved in early embryonic development. And we hope to be able to translate these findings into treatments that can help solve birth defects in the population. Thank you.